Hi guys, today we're going to learn about how things move in and out of the cell. There are two main categories of cell transport. Today we're going to talk about passive transport and later we will talk about active transport. So when you think of passive transport, my stupid science analogy to help you remember this is think of a mountain. On a mountain you have your high side and your low side. Now we're going to imagine this means high concentration of particles versus low concentration of particles. And then we're going to add some water to our mountain, which we'll talk about more in a little bit. Now, if I was to put somebody in a boat at the top of my mountain, would they have to paddle much to go down the mountain? No, they wouldn't, right? They're going to go from high concentration to low concentration without having to paddle. They wouldn't have to do anything. They would just naturally flow. So there's no energy needed. This is what passive transport is the movement of particles from high concentration to low concentration without the use of any energy. Active transport is a little bit different. This is if I put you in the boat at the bottom of the hill, or low concentration, and told you to make your way up the hill. In order to go from low concentration to high, you're going to have to do a lot of paddling. This would take a lot of energy, or you'd have to be super active to do this. So passive is going from high to low with no energy, and active is going from low to high using lots of energy. Now the water's there for a reason too, because the water actually moves passively as well, but it has its own little name for that. So water on your hill is going to go down the hill from high concentration to low. And this is called osmosis, which is one of the kinds of passive transport that we're going to be talking about. So remember, all of these things in real life are not going down a hill, but actually going through your cell membrane. Right? Cell membrane is made out of proteins and lipids called phospholipids, and you can see these here with the phosphate heads and the fatty acid tails. The purpose of your cell membrane is to control what enters and leaves the cell in order to maintain something called homeostasis. In science word, homo means the same. So homeostasis is keeping the same internal balance within your cells or within your body. And it is the membrane that allows cells to do that. So here's passive transport. Like I said earlier, it requires no energy. You're going from high concentration to low. It does need a concentration gradient to begin with. And remember, that's the difference in the quantity of molecules over a space, regardless of what kind of molecule that is. And there are three different kinds of passive transport that you are responsible for. There is simple diffusion, facilitated diffusion, and then osmosis. So let's get into the different kinds. With regular old diffusion, this is the net movement of small particles from an area of high concentration to an area of low concentration until they reach what's called equilibrium, when everything is equal. This is what oxygen does through our membrane when it flows in right through the phospholipids. If your cells are low in oxygen and there's oxygen that is dissolved in the extracellular fluid, it will flow right in. This is also what happens when somebody farts in class. When somebody farts in class, there is a high concentration of what we'll call poo particles, right at the person who really has flatulence. All right, those molecules are not going to stay there. They travel from high concentration down to low steadily. So those who dealt it, smelt it is true that there's a high concentration there, so they're going to be the ones that smell it first. But slightly thereafter, the next person next to them is going to smell it. And then eventually it will travel throughout the whole classroom. Now, if you're far enough away from the person who dealt it, you may not land up smelling it, because by then the amount of atoms, poo particles we're saying, will be so little that it won't be enough to trigger the nerves in your nose to say, hey, this is an unpleasant smell. So diffusion is how our poo particles move. It's also how our oxygen and carbon dioxide moves inside our cells. I know, your parents probably think your teacher's super weird now. So here's an animation of diffusion. Your molecules are moving from high concentration to low until they are equally dispersed. That's equilibrium. Here's another picture of diffusion where I have a high concentration and this dotted line is like a semi-permeable membrane. It's going to let some things through and not others. In this case, the red solutes, which is the thing dissolved, cannot fit through this membrane, but water can, because water is only 3 atoms big, H2O. It's very small. So what will happen is the water will leave this low concentration side and go into the high concentration side, trying to water this down until everything is equal. Now, if the red molecules could fit through the membrane, they would just travel until there's equal concentrations again. So either the solutes move or the water moves, depending on what the membrane is doing. So this equilibrium, its full name is dynamic equilibrium, means there are equal amounts throughout. But these molecules are not standing still. They are still moving back and forth through the membrane. They're just doing it at an equal rate, so there's no change in the cells involved. But there are things that can affect the rate of diffusion. All right, concentration is definitely one of them. So if we go back to our flagellants example, if one of your classmates has been eating a whole bunch of bean burritos and comes into class and does a giant man-sized bean burrito fart, that fart is going to travel faster and farther than like a tiny little dainty girly poof, okay? So concentration plays a big role. If you think of it like baking cookies, if you have a mom who's nice enough to bake you cookies, if mom is baking 4 dozen cookies, versus just 1 dozen cookies, the smell of the cookies baking is going to go farther than if she's only doing a couple cookies. Temperature also can affect the rate of diffusion. So if you're talking about flagellants again, hot flagellants travels farther than cold flagellants. So if you are in a hot room, your fart is going to move faster and it's going to spread farther than if you're outside on a cold day. So if you're trying to hide it, go into a cold place, it won't travel as far, but then it's going to stay where you are. So you got to walk away and leave it there. All right, pressure is another thing. You can't really put your farts under pressure, but you sure use this whenever you use anything in a spray can. So the Lysol or air freshener you're going to use after somebody has flagellants, you're going to press the top of the can, and that is gas under pressure that once you release the pressure, it comes shooting out of the can very rapidly. That is still diffusion, it's just under pressure. And that's why after you've used your can for a while, you might still shake it and feel some liquid in there, but if that liquid is not under pressure, it won't squirt out anymore. Now, facilitated diffusion is a little bit different. Facilitated diffusion is diffusion with a helper. To facilitate something means to help something occur. So this happens when you're moving larger molecules like glucose, C6H12O6, and you got to get through that membrane. So this is one of your proteins that are going to help this occur, and in particular your carrier protein that we talked in class about before. So the molecule will dock in the carrier protein, and the carrier protein will change shape to drag that molecule across. It does not require energy because you're still going from high concentration outside of your cell to low concentration inside of your cell, but it does require a protein helper. Then we have osmosis. Osmosis is the net movement of water from an area of high concentration to an area of low concentration. The only difference between osmosis and diffusion is small molecules and osmosis is specific to water. And you may have seen an old movie called Osmosis Jones. Osmosis Jones was blue colored because he was water. So what water does is, here's some solutes again. These solutes are too big, it's sugar in this case, too big to fit through the cell membrane. Because the solutes will not fit through, the water on this side that has no solutes is going to flow to the other side trying to reach a 0% concentration of the sugar, which it never can. So the water will keep pushing through that membrane, defying gravity and raising the level on this side. All right? So water does this quite readily. If you think about your bathtub at home, when you fill your bathtub, the water's all falling on one end of your bathtub, and as long as your bathtub is plugged and it starts filling up, it's not like that one end of your bathtub is the only end that fills. It goes over to the other side of the bathtub as well to reach equilibrium. So here's another example with a selectively permeable or semi-permeable membrane, where on the right here we have a higher concentration of solutes, and the solutes are too big to fit through the membrane. And on the left we have less solutes. So the water is going to go towards the more solutes to try to water this down to be equal with the others. And that'll do two things. It'll concentrate this side a little bit more and make this side a little less concentrated until they are equal. And it pushes against gravity to do this. You must have a semi-permeable membrane though to make this work. Otherwise, your solutes would just equalize out too, and then everything would be level. So this brings us to some new words and how these things move with osmosis. We have tonicity is what this has to do with the movement of these molecules. And this term isotonic. Iso means the same. So isotonic is when you have the same amount of solutes in the cell as outside of the cell. So if you look at this picture, I got 10% salt in the cell and 10% salt outside. They are the same. Okay, the salt is the solutes, and it can be any kind of solute. It does not matter. It's just the stuff dissolved into water. So if it's the same, these molecules are going to move in and out equally, and there's going to be no change in your cell at all. It's going to stay the same. So a good example of this is your red blood cells in salt solution. When you go to the hospital and you need fluids, they don't give you plain water. They give you saline solution. Saline solution is 0.9% salt, and that's because your body has 0.9% solutes in it. By both of them being 0.9%, it makes it isotonic to your blood cells, and everything stays the same. The fluid goes in your body without doing any damage, which is what you want. Another example would be putting seaweed in ocean water. So seaweed naturally has a lot of solutes in their cells. And the reason why they have that is because they need to be isotonic with the ocean water that has a bunch of salt in it, and that's solutes. If seaweed did not have all those solutes, they would not be isotonic, and something very bad would happen to the seaweed. So here's when things could go a little bad. A hypotonic solution. Hypo means less, like hypothermia means you have less heat in your body, all right? So hypotonic is less solutes. It's all about the solutes. Will tell you where the water is going to go. So if you look inside my cell here, my cell has 20% salt. That is not less than 10%. So out here in the beaker where I have 10% salt, this is my hypotonic solution because it has less, okay? And our rule is water always moves towards the most solutes or out of the hypotonic solution. But if you know one of those, you know the water moves towards the most solutes, you can always predict where it's going to go. So in this case, the water is going to move into my cell and start watering down this 20% trying to reach 10. In the process, if you have a whole bunch of water going into your cell is going to expand. Think of it like a water balloon, right? Empty water balloon, you start pumping water in there, it's going to blow up. And then if it gets too much water, it could pop, all right? And that is bad news. So if we took your red blood cells and you put them in pure water has 0 solutes. Well, if that happens, all that water is going to rush into your red blood cells and then pop, 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 they pop. There was a real life example of this when a lady who had joined in a contest that was a radio contest when the first Xboxes came out, a long time ago when I was a student teacher. And so what the contest was is to drink the most gallons of water without throwing up. And she really wanted to win this Xbox for her kid, and so she signed up. Now, the radio DJs did not have a basic understanding of science. And so when they went to the grocery store to pick up water, instead of picking up regular old drinking water, which naturally has solutes, they picked up what's called distilled water. Distilled water has 0% solutes. Because it has 0 solutes, that means that the water started rushing into her cells as soon as she started drinking it. And she was drinking gallons of this stuff, right? So she's drinking gallons of gallons of distilled water, and somehow was able to not vomit, because in most cases you would start to vomit, but she was able not to. And when she was drinking all this water, at first it was fine, but then her cells started filling up. And as they filled up, they started popping. And the cells in her organs started popping until her organs failed and she died. She died all because the radio DJs did not understand the difference between drinking water and distilled water, which is pure water, okay? You can't expose your cells to that without damage. So that's like if you took a saltwater protist, which is a little organism like an amoeba, or you could even think of it as a saltwater fish. If you took a saltwater fish and you placed it in fresh water, then your fish would die very, very quickly. And what you're doing internally to your fish is popping their cells. Now, the opposite of hypotonic is hypertonic. Hyper means more, like if you're hyperactive, you have more energy. So this is when you have more solute. So if we look at our cell here, outside I have 20% salt. This would be hypertonic, it has more. And inside, where I only have 10%, this would be hypotonic. They're always the opposite of each other. Hypertonic, the other one's hypotonic, because one's more, that means the other one is less. And so our rule, if you remember, is that water always moves towards the most solute. So water is going to leave my cell and go out into the solution, trying to water down this 20% to get closer to 10. Now this is not going to work out so well for this cell. This cell is going to shrink, and then eventually it would die because of that. So it removes the water. So this in this case it's salt, which most of us can relate to the fact that salt will pull water out. That's why if you're ever stranded on a desert island, you can't drink salt water, because the salt water will actually dehydrate you more by pulling water out of your cells, and then you will die quicker, right? So it could also be sugar. Sugar does the same thing. And if you think about soda, soda has the double whammy of a bunch of sugar and some salt dissolved in it that's going to pull water out. So soda is not the best thing to hydrate yourself, all right? You really need some regular drinking water for that. So an example of this is if we took our red blood cells and we put them in a 10% salt solution. So remember, saline is only 0.9 at isotonic, when it's isotonic with your blood cells. So your blood cells have 0.9, salt solution's 10%, water's going to go towards the most solutes. The water is going to leave your red blood cells and go into the salt solution, and your red blood cells are going to shrivel right up. All right, you also can think of a freshwater fish or protist. If you take your freshwater fish and you throw it in salt water, it's going to die real quick, because that salt water is going to pull all the water out of that fish's cells, and then they die of dehydration. So this is what that looks like. So when you put your red blood cells in a hypertonic solution, essentially what's happening is that the water is leaving your cells and going into the solution, which makes your red blood cells shrivel up. And once they shrivel up, they are no longer carrying oxygen for you, and that is bad. If it's isotonic, that means they're the same, both 0.9%, the water will go out equally in and out, and there will be no change in your blood cells. And then if you're in a hypotonic solution, the water will rush into your cells, and then they will fill up, and then you can see this little one over here has popped, okay? So hypotonic, less. Isotonic, same, in and out. And hypertonic is more, so in this case the water will come out, and I didn't do the arrow over here. Hypo makes it go in. All right, so always towards the most solutes. Here's more solutes, water's going towards this. Same solutes, in and out. Here's more solutes, water's going towards that. Okay. So in plants, it's a little different. The whole cell does not shrink in plants, because plants have this rigid structure that surrounds the cell called the cell wall. So plants do this thing called plasmolysis. There's that lysis word yet again that means to break. Plasmo for the this plasma membrane in the cytoplasm, right? All that stuff. So when you pull water from your plant cell, you put this in salt solution, the first thing that loses water is your vacuole, because your vacuole holds extra water, food, and waste. And in plants, it's mostly water. So first you empty your vacuole, and you can see this guy's already shrinking over here. Then you pull water out of your cytoplasm, because remember, your cytoplasm is mostly water. And so your cytoplasm, along with your cell membrane or plasma membrane, shrinks up. But your cell wall is rigid and protects, and so it stays the same. Oh, my line got a little wonky there. All right, it stays the same, and what happens when this occurs is your plant will wilt if you pull the water out, right? If you don't water your plant, it wilts, because this vacuole, when it's full, creates pressure against the cell wall to help stand your plant up. And when that is all empty, your cell walls start getting a little wobbly, and your plant starts to droop. So turgor pressure is the name for the opposite. This is when you water your plant, when water is going into that vacuole and it fills the vacuole up. Remember, the vacuole fills up. Think of like a water balloon in the center of a box, okay? The water balloon fills up and it pushes against the walls of the box, and it makes that box a little more sturdy, okay? That's turgor pressure. You're creating pressure by increasing the water. Plasmolysis is the opposite. This is when the water comes out because you're in a hypertonic solution, and then your plant wilts. All right, and that's all about that vacuole. So to review, passive transport requires no energy to go from high to low concentration. 3 different kinds are diffusion, which is the movement of small particles right down that concentration gradient. Osmosis is the movement of water molecules down your concentration gradient. And facilitated diffusion is the movement of large molecules with the help of a protein, also down the concentration gradient. All right, guys, see you in a little bit.